We conducted research on the expression of appetite-regulating genes in avian models by Nora Hagigi, Norley B. Croft, and Ali McCullen.
The information known prior to the experiment was that the hypothalamus regulates appetite by signaling the peripheral and central nervous systems. The most involved areas associated with appetite in the hypothalamus are the lateral hypothalamus, known as the LHA, paraventricular nucleus, known as the PVN, ventromedial hypothalamus, known as VMH, dorsomedial nucleus, known as DMN, and the arcuate nucleus, known as the ARC. The main area involved in energy usage is the hypothalamic melanocortin system and the ARC. The corticotropin releasing factor is one of the most abundant neuropeptides associated with genes involving anorexia in the chicken. Although genomes in relation to the hypothalamus have been studied extensively, there has not been much research on patterns of appetite. In efforts to discover contrasting factors, appetites present in Virginia low, known as LWS, and high, known as HWS, body weight lines of chickens, it is necessary to measure amounts of mRNA present in the hypothalamus of each independent variable. Conducting this experiment is important because, through examining gene expression in the individual nuclei of the LWS and HWS chicks, comprehension of the neurocircuitry in the hypothalamus that relates to appetite can be better understood. We can also gain further knowledge of the differences between LWS and HWS chicks as far as appetite regulation, which we did through measuring mRNA expression profiles of appetite-related components in individual nuclei, comprised of the ARC, DMN, LHA, PVN, and VMH, during both times where the chicks were both fed and fasted. Additionally, the experiment is important because upon investigating the genetic makeup of appetite regulation in chicks, the makeup can be related to that of humans to help us further understand appetite-related diseases such as obesity and anorexia as they manifest in humans. This figure is an overall depiction of the hypothalamus in a chick. Right here is a main structure found in the hypothalamus called LHA that stands for the lateral hypothalamus. This structure is the EPD, which is also known as the paraventricular nucleus. The section depicted here is the DMA, which is also the dorsomedial nucleus. The two structures from the hypothalamus depicted here are the ARC, which is known as the arcuate nucleus, and the VMH, which is also known as the ventromedial hypothalamus.
The chickens used in this experiment are from lines that have been long-term selected for low and high body weight. The original chicken population was comprised of crosses of 7 inbred lines. Upon chicks being hatched, they were group caged for 1 day and then transferred to their own cages, and all were provided with a MASH diet. 5 days post-hatching, chicks were divided into 4 groups at random. The groups included LWS fed, LWS fasted, HWS fed, and HWS fasted. Chicks were then given anesthetics through cardiopuncture and were then perfused through the carotid artery with RNA stabilizing buffer. After perfusion, coronal sections were collected in a chirostat. However, only punches that were confirmed for location accuracy were used for total RNA isolation. After this entire process, mRNA prevalence was measured in appetite-associated areas.
Table 1 shows the primers used for real-time polymerase chain reaction, whereas Table 2 shows the mRNA prevalence in the DMN.
After conducting the experiment, we came up with some interesting results. Neuropeptide Y presence in the DMN was more prevalent in LWS versus HWS chicks. The impact of MC3R was heightened by fasting in LWS. However, the same results were not present in HWS. Instead of the chickens who were fed, the chickens who fasted CFOS was more abundant, and the expression of NPY receptor subtype Y was more abundant in the fasting state in the LHA. The chickens that underwent fasting expressed mesotocin receptors in the LWS and HWS. However, not as much in the LWS that was fed. NPY receptors in orexin presented themselves heavily in the LHA of the LWS instead of the HWS. In HWS, NPYR1, MC4R, which is agonist and antagonist of melanocortin receptor 4, and CART, which is amphetamine-regulated transcript, was more prevalent in the PFN, which contains periarcha that synthesizes oxytocin and corticotropin releasing factor, which is known as CRF, compared to LWS. In LWS, NPYR5 and mesotocin MT was greater here than in the HWS. When the chickens who were fed and the chickens who fasted were compared, NPYR5 and ghrelin mRNA was found in higher quantities. When observing the VMH, it was found that LPR and orexin receptor 2 was found to be greater in LWS and HWS in the fasted chicks, as well as CFOS abundance being greater in the fasted chicks. In the chicks that were fed, NPY2 mRNA presented itself more in the ARC. In LWS, NPY was generated more due to fasting, but this did not seem to be the case in the HWS chicks. CRFR2 and NPY had an interaction of line and treatment. Fasting appeared to have a great effect on expression of CRFR2 in HWS chicks instead of LWS chicks in the ALC. In the ARC, NPY was found in high quantities from fasting in LWS versus HWS chicks. Due to the p-values being less than 0.05, all of these results were found statistically significant when analyzed by ANOVA.
Here we have our breakdown of the mRNA abundances in the hypothalamus. Performing this experiment with chicks only 5 days of age was vital because some LWS chicks die soon after the absorption of the yolk sac. This is where severe anorexia can only truly be researched at this age. Examining the results of this experiment is hard due to the baseline amount and expression patterns of appetite-associated factors within the brain being unknown. Another difficulty we ran into is that the regulation of appetite is complex with the copious amounts of pathways involved in appetite regulation at the same time. Therefore, it was difficult to pinpoint exactly which regions played a vital role in the appetite regulation process. The results we found in this experiment give vital information about the regulation in perikarya, which is essentially the cell body of the neuron that contains a nucleus, which contains mRNA. These results are also specific to the location of where the mRNA is within the neuron. Usually, the site of the neuropeptides and where they are released are far from one another. The VMH is the main center in the hypothalamus for activities taking place in the neuron associated with appetite. Neuropeptide Y is one of the most examined neuropeptides in relation to appetite due to its high presence in the cell across various species. The ARC is highly studied due to it being one of the most commonly understood structures involved in appetite regulation. The reason NPY is important to this study is due to the original report stating that LWS is non-responsive to food intake when NPY is present. Overall, the LWS chicks demonstrated a high concentration of ORXR2 present in the VMH, orexin in the LHA, MT in the PVN, and greatest abundance of NPY was found in the ARC and the DMN. All of these things lead us to believe the NPY signals are present in the LWS chicks, but due to the presence of the anorexigenic nature, the NPY was overshadowed. Due to the higher amounts of MC3R in the nucleus and AGRP in the ARC, illustrates the melanocortin signals in the LWS chicks is of more significance than in HWS chicks, which can also play a role in the physical makeup of the anorexic chicks. The chicks who were fasting demonstrated few differences in higher concentrations in the DMN, MC3R, and ARC NPY in LWS chicks, but this was not the same scenario in HWS chicks. Uncontrollable feeding activities in obese chicks can be conducive to increased amount of CART in the PVN of HWS versus LWS chicks. Also, elements that appear between the lines can be associated in neural pathways involved in appetite regulation, including NPY, melanocortin, orexin, and mesotocin transmittance. Overall, these neural pathways can be a factor in anorexia present in LWS chicks, as well as give us insight into how total mass of fat of an animal can contribute to the overall contrasting appetites in the chicks. The following tables and figures show the results of the experiment and helped us further understand the extent of our finding. Tables 3 and 4 show the mRNA prevalence in the LHA and the PVN. Tables 5 and 6 show the mRNA prevalence in the VMH and the ARC. Figure 1 depicts LWS and HWS and their respective abundances in the hypothalamus. Based on the research we did, we had a few critiques, and we would like to know more about the environment in which the chickens who hatched the chicks were raised in. Also, we would like to know why or how this is applicable or translational to humans, if at all. We also would like to increase background knowledge on the importance of the structures in the hypothalamus that are discussed and why they play such a vital role in appetite. Also, I know it is hard to allow chickens that are anorexic to reach a point beyond adolescence, but in additional studies, potentially observing adult chickens with these attributes may be beneficial because over time, mRNA can change.